Joining us tonight are Dr. Given Harper and Stephen Burkett. They will provide us with updates on some research projects they've been involved with. Some of the questions that their research projects attempt to answer are, how can one determine the sex of captured red-tailed hawks? At what latitude did they spend their previous summer? And where do the subspecies of the hawks winter in Illinois? A little on both of our speakers here. Dr. Harper is the George C. and Ella Beach Lewis Endowed Chair of Biology at Illinois Wesleyan University. His areas of interest are centered on various aspects of avian ecology and wildlife toxicology. Giving the presentation tonight will be Stephen Burkett. Stephen is a senior environmental studies ecology concentration major at Illinois Wesleyan. He will be graduating in a couple weeks in May. In addition to working on these research projects, over the past 2 summers, Stephen has worked to restore native habitat for the Parklands Foundation in central Illinois, where he conducted restoration work with an ecological consulting firm in St. Louis. Following graduation, Stephen plans to work with the Wyoming Conservation Corps in Laramie before attending graduate school. If anybody has any questions on the presentation, please save them for the end. We'll have some questions. We'll have some time for questions at the end. So with that, Stephen, if you want to share your screen and start the presentation, you're on.
Awesome. Thank you very much for that intro. I'll get my screen shared here. All right. So yeah, as Robert had just said, my name is Stephen Burkett, and I'm going to be giving you guys some updates on the studies of wintering red-tailed hawks in Illinois. A little bit of an overview about the coming presentation. I'm going to start with a bit of a biography, a little bit about me, why I'm here talking to you guys today, and then we'll go into an introduction about the basics of the red-tailed hawk, and then following that, a little bit of how we collected our data on these red-tailed hawks. After that, we'll talk about our 3 different projects, as mentioned earlier, the in-hand sex determination, so looking at how we can determine the sex of these birds when we've had them captured, followed by the summer latitudes of wintering red-tailed hawks in Illinois. So trying to figure out where captured hawks that have been wintering in Illinois spent their breeding season. Following that, we'll talk about the subspecies distribution of wintering red-tailed hawks in Illinois. So a little bit about me. Big into birding. I've been birding since long before I can even remember, and got really into it kind of my sophomore year when I was invited to join Dr. Harper on the spring and Christmas bird counts, and as well on raptor surveys in McLean and Tazewell counties. So the winter raptor surveys for that. And as well as mentioned earlier, I'm big into ecological restoration, working to restore native prairies and natural areas. I got to work for the Parklands Foundation in central Illinois, and as well in St. Louis, working to implement green infrastructure and restore native habitats there. With that job in St. Louis, I actually also got to work in Columbia, Missouri and Kansas City, Missouri, where I did a lot of work on roadside prairie restoration and working to remove honeysuckle, invasive honeysuckle from the forest preserves in Kansas City. Another thing that I'm very interested in and spend a lot of time trying to and get better at is something called digiscoping, which I'm sure many birders are familiar with, right? You just using my iPhone and kind of my binoculars or a spotting scope, I'm able to take pictures of wildlife and especially birds. So on the left here, a prairie merlin that I actually saw with Dr. Harper on a raptor survey in McLean County. And in the middle there, an American goldfinch that I took a picture of in my hometown in St. Louis. So a little bit of an introduction to the red-tailed hawk. Buteo jamaicensis, these are one of the most common diurnal raptor in North America. So they have done actually pretty successful following human urbanization. They can be found in rural, suburban, and even urban areas in Illinois. Often seen soaring over fields or perched on utility poles near roads. They really enjoy kind of those grassy regions on an interstate or on a side road where they can look for kind of their prey, their small mammals and that kind of thing. Some easily identification and diagnostics for the red-tailed hawk. They're bulky birds, and they soar with their broad rounded wings and short wide tail. One of the kind of key diagnostics to red-tailed hawks, if you can see where I'm pointing here on this upper image, is the dark upper patagial marking. So kind of on their upper wing, they'll have typically a nice dark mark there. And that's a good way to tell that you're looking at a red-tailed hawk, right? In the adults as well, the characteristic red tail, it begins to be developed after their second year. And that's different than the juveniles, who will have sort of a brown tail with some heavily marked bars typically. And so that's how you can sort of tell an adult versus a juvenile red-tailed hawk. Again, they feed primarily on small mammals, looking in those grassy areas and hunting for voles, moles, mouse, rabbits, anything they can get their talons on. And so they'll use their strong talons, and they're very adept hunters to catch their prey and tear it apart with their sharp curved beak. A little bit about red-tailed hawks, their movement and their subspecies, and as well as the differences between the sexes. So red-tailed hawks are a migratory species where they migrate short to intermediate distances. However, many regions of the United States and portions of Canada have year-round residents. Illinois is one of those places, and we have one such subspecies that spends their year-round time, the eastern subspecies, borealis, in Illinois. A subspecies, as I've just mentioned, is a geographically isolated population of these red-tailed hawks. And in the example of red-tailed hawks, it's really interesting. They have developed these super distinct plumage variations between the subspecies. And we can actually see that here on the right. So a super dark Harlan subspecies up here in this first picture, followed by a much lighter borealis. There are 14 recognized subspecies of red-tailed hawk, 4 of which are known to winter in Illinois. However, there's one putative subspecies, meaning scientists and ornithologists, the gig is still up. They're still trying to decide if this is a purely new subspecies or if it's just a different plumage variation of these red-tailed hawks, a darker northern subspecies. And we'll talk more about that and get to see more into the subspecies a little bit later on here. And then finally, about the sex differences between red-tailed hawks, so between males and females, I always find this interesting. You know, when we're thinking about passerines or maybe some songbirds like a northern cardinal or indigo bunting, the males are vibrant and bright and colorful compared to a drab female. So that really makes determining sex easy in adult breeding birds. However, red-tailed hawks specifically, and some other raptors, do not have this plumage variation between males and females. And so really the only way you can distinguish a male red-tailed hawk from a female red-tailed hawk is that females are typically a quarter to a third larger than males in mass and body size. Other than that, there are no distinctions based on looks. And so these are kind of 3 main things to keep in mind when talking about our projects and why they're important. We'll get into how we collected data for our 3 projects here. So essentially, we had to live trap red-tailed hawks in order to collect both feather samples and measurements that we'll get into here in a second. Our trapping took place in the winter months. So we trapped from December, January, and February. We started in December of 2020 and ended our, 3 trapping seasons this February of 2023. So we spent 9 months total trapping and caught a total of 95 birds, which is pretty successful. McLean County, that's where we kind of was home base for us, is where Illinois Wesleyan is, as you can see right here in this blue central region. And that's where a majority of our birds get sampled from. We caught a lot of birds in the central region there, the blue region, and a lot of birds as well in the DuPage, Cook, and Lake area from Chuck Rizzo, who we collaborated with and was able to catch several birds and increase our sample size and our region and distribution to the north of where we're getting birds from. Southern Illinois was somewhat difficult to get to just based on distance, and we did catch several birds in the orange kind of southern region, but not nearly as many as in the central or northern portions. So how do we actually catch a red-tailed hawk may be a question kind of in your mind, but what we use is a balsa tree trap is what they call it, and that's right here in the middle. It sort of blends in with the grass pretty well, but what it is a double layer of metal wire that's kind of a cage where we can put a live mouse inside with some food and enough room for it to run around and make some commotion. And around that metal cage is actually monofilament fishing line tied into nooses that can get tangled around the hawk's talons and feet when it comes down to try and catch the mouse. Now this trap is weighted, and in order to keep the hawk from carrying our mouse and our trap away and keep the hawk safe and from dragging it into the road. So in order to put the trap out, what we would do is drive around Illinois and look on these rural, slow, low traffic roads for a red-tailed hawk that's perched in a tree or in a telephone pole. And essentially we would make our approach, put the trap on the side of the road far enough into the grass where it's nice and safe for the bird, and there's no possibility of being interfered with by pedestrians or bikes or cars or anything of the such. That safety with the bird was really a main priority when conducting all this research and making sure that no injuries occurred, or if there were any cuts or scrapes or anything of that matter, we put triple antibiotic on it and made sure the bird got the best care possible. Once we caught a bird though, however, we wanted to be as quick as possible of getting it out of the trap and getting it unstuck. And so we would rush over and quickly put on what we call a falconer's hood, which you see over here. And this is calming the bird down, making it much more docile and easier to work with when trying to take our measurements and collect feather samples. Down in the bottom is sort of a nice picture to represent the materials and stuff we would bring out when we went trapping, bring a backup trap, extra measuring utensils, a scale. This shoebox, interestingly enough, is where we would put the cage for the mice when they would be together and not in the trap. So they stayed nice and warm and in a dark environment to keep them calm. And then finally, interestingly enough, this Folgers coffee can, if you see here, is 2 Folgers coffee cans taped together that we can put the bird in and keep it nice and calm and not from wiggling around when we put it on the scale to take the weight. So once we've caught our red-tailed hawk and we have one and we're ready to collect measurements from it, that's the first thing we do, we collect measurements. So we start usually with the wing cord, and that's this measurement here. Sort of starting from the patagial marking, there's a bend in the wing, and we'll measure down to the longest extending primary feather. We also take the mass, a measurement of the tail, something called the culmen, which is essentially just a beak measurement. We're measuring from the fleshy part called the cere down to the tip, and then the hallux, which is a measurement of the rear talon. Another method of collecting data from every one of the red-tailed hawks that we caught was we took feather samples. So we took 2 different types of feather samples from each of these red-tailed hawks, the first of which was a breast feather sample. So we collected 3 plucked breast feathers from each bird. And the idea behind plucking the feathers opposed from cutting them or any other method is to keep a little bit of the skin cells on the base of the feather where it connected to the body of the bird. And that's critical for when we want to try and collect DNA from this sample, because the skin cell is going to have more DNA retained in it than, say, the keratin of the feather. And this is a much less invasive method of DNA analysis and DNA collection than, say, a blood sample, and is relatively easy on the bird. Another method we collect is the primary feather. And so if you'll see here, the primary feathers are counted out, and we collect from the very first primary feather. And this is important because this first primary feather is the first feather that's grown in on the breeding ground of these red-tailed hawks. So after they breed, they molt their feathers and regrow this first primary feather in first, and then it works outwards. So this is going to be an important idea to remember when we're discussing our summer latitudinal origins paper that looks exactly at this feather sample. So our first project is looking at how we can determine the sex of these red-tailed hawks in hand. So as we mentioned earlier, there's no visual differences between sexes, and essentially the only way to accurately determine the sex of these red-tailed hawks is through DNA analysis and invasive field cloacal examinations, essentially, which are highly invasive on the bird and not very comfortable, essentially. So what researchers have been looking to do is create equations using these measurements to accurately sex red-tailed hawks. Donahue and Dufty in 2006 were able to highly accurately sex adult and juvenile red-tailed hawks using samples of Western birds. So that's a bit of a problem for us as we're on the east side of the United States, and we have primarily the Eastern red-tailed hawk subspecies here. Washburn and others recently in 2022 developed equations specifically for these eastern red-tailed hawks. However, their accuracy was slightly lower likely due to their larger sample size or the robustness of their equations. So we really wanted to look at how these equations can be applied to our sample and see what sort of accuracy we could come up with. We used the morphological measurements gathered from most of our live trap birds and as well as frozen red-tailed hawks to test the accuracy of these equations. We were only able to use and collect and get solid DNA analysis run on 68 of the feathers just due to the sampling process, and we only started collecting the breast feathers in our second season of trapping, so that's why it's a little bit lower than the 95 total sample size. The frozen specimens we gained from the Illinois State University Museum, and we used 22 of them in our sample. So to determine the sex of these red-tailed hawks, we have to use something called PCR analysis, right? And essentially what we're doing within this PCR analysis is extracting DNA from those feathers. And we're trying to get the DNA out of the skin cell at the end of that feather sample. So what we do is cut a very, very small portion of it, about 2 millimeters, and hopefully that portion of the feather has a bit of skin attached that we can put in a solution and essentially induce lysis, which is a controlled explosion of the cell, if you will, that kind of puts this DNA in solution. So once this DNA is in a solution, we're able to put it through the PCR kind of amplification process and replicate a specific region of that DNA to nearly 1 billion times. And once we have this highly, highly concentrated section of DNA from that specific bird, we can then put it into a process called gel electrophoresis, which essentially is taking the negative charged DNA, running an electric field through it, and pushing it through this gel that we can see on the right. And so what we're actually able to see here is the two different sex chromosomes of these red-tailed hawks. So in these birds, a female is going to have both a W and a Z chromosome, and males are only going to have 2 Z chromosomes. And so the Z chromosome is a longer chain of DNA, and thus is a heavier kind of molecule there, and is going to not travel as far down the gel through this electrophoresis process. And so that's why we see with a male subject here that only has 1 bar and is higher up than, say, the female to the right here that has both a W and a Z chromosome shown. This sample over here to the far right is a little bit easier to see the 2 bars, but the reason that W chromosome is visualized lower is because it's a shorter chain and thus lighter. So once we have the sex of our red-tailed hawks, our live trap red-tailed hawks determined, we then are able to plug our measurements into different equations. So we used the Washburn equations and Donahue and Dufty equations and plugged in for both adults and juvenile birds. So this was the first study to test the Donahue and Dufty equations on the eastern red-tailed subspecies of red-tailed hawk. And we actually saw success in accuracy in both the adult and juvenile tests. You can see the Donahue and Dufty is in the blue and the Washburn et al. Is in the green. This was also the first study to test the removal of emaciated individuals from results. And so essentially emaciated birds are just lighter, malnourished, and don't have quite the fat deposits of a healthy red-tailed hawk. This is influential in our kind of study because when we're doing these equations, if the predicted or the known sex of the bird is a female and we plug it into the equation as it's emaciated, it may predict the bird is actually a male. So it can throw off the accuracy a little bit in that way, and that's why we decided to remove those individuals from our testing. Another thing we did with these equations was we actually modified them. So we modified the constants in sort of the wing and mass measurements there, and both the Washburn and Donahue and Dufty equations. And this was also, we found significant increases in our modified adult equations compared to the traditional Washburn and Donahue and Dufty equations. So for Washburn modified adult, we saw a 20% increase in accuracy. And for the Donahue and Dufty modified adult, we saw a 14.5% increase in accuracy, which is significantly increased. And that's one of the reasons you don't see a juvenile modified equation is we couldn't find a way to consistently improve and significantly improve the accuracy of the juvenile equations for both Donahue and Dufty and Washburn. So that's a little bit about the sex determination project, and now we'll go into determining the summer latitude of wintering red-tailed hawks in Illinois. So a little bit of an introduction here. Paprocki and others in 2017 suggested that red-tailed hawks have undergone substantial changes in migratory behavior over the past 30 years. Their hypothesis is that this substantial change in migratory behavior is possibly due to climate change. And they hypothesize that fewer red-tailed hawks are migrating in the fall due to climate change. So our study is looking to test this hypothesis, and we are looking to see if there are more year-round residents in Illinois, or if red-tailed hawks are still migrating in the fall, like traditionally. So to do so, we have to use something called hydrogen isotopes and an Isoscape map to predict where these red-tailed hawks that were wintering in Illinois spent their breeding season. So to do so, we use something called hydrogen isotopes, and an isotope is just a version of an element that has a different chemical weight, essentially. So you see here the 3 different versions of a hydrogen isotope. These will start with the original and slightly get heavier as you go around. So the different hydrogen isotopes have different chemical weights, and thus are different heavinesses, correct? And so essentially, when this is implemented into water in H2O, it lays out within precipitation on a gradient across the United States or across North America, right? So starting here in the red, you'll have a higher concentration of the heavy hydrogen isotopes. And as you go north, it'll slowly become more concentrated on the lighter hydrogen isotopes, so the protium and that kind of thing. And so this actually helps us as the precipitation builds up through the food web and into the red-tailed hawk's feather. We can use the hydrogen isotope ratio in the feather to predict where it's going to fall on this map. And so since that first primary feather is the first feather to grow back on the breeding ground, we can then compare this map to where that feather is located and determine and predict highly accurately where that bird is coming from. So to collect the hydrogen isotope ratios from these feathers, we have to go through a very specific process of feather cleaning and essentially remove any possible abnormalities on the feather or anything that could throw off the hydrogen isotope ratio that's not purely from the feather. So we want to make sure the feather is as clean as possible, as pure as possible to get the most accurate ratio. These samples are then sent out to the University of Wyoming Stable Isotope Facility, where they can be analyzed for their hydrogen isotope ratio, the ratio of those heavy hydrogen isotopes to the lighter ones. So then what we can do is create an Isoscape map based on hydrogen isotope ratios in adult and juvenile red-tailed hawks of known breeding location, and compare that to the hydrogen isotope ratios from live trap birds that we trapped in the winter months. And so comparing the birds that we know where they spent their winter based on the hydrogen, or spent their summer based on the hydrogen isotope ratios, and comparing that to the ones that we live trapped in the winter, we're able to get a sense of where these birds spending their winter in Illinois spent their summer and are breeding at. And that'll give us an idea of if they're residents or if they're migratory birds, right? And so essentially we'll get something that looks like this over here to the right. This was a study done by Taylor Duray on American kestrels. And what you can see is this blue dot in the middle is where the American kestrel was live trapped and taken the feather sample from. And the blue region is the predicted area where it spent its breeding season and where it spent its summer. So we'll be working on trying to create these style maps with our collected winter samples, and we still need to create that calibration function of known breeding locations to be able to do so. So that's kind of one of our next steps. We've already got some samples sent out to the lab and came back, but we have some more to clean and a few more steps to get totally done there. So the final project that we've been working on is looking to determine the distribution of subspecies of wintering red-tailed hawks in Illinois and give kind of updates to those maps. So the methods for this project, essentially collecting subspecies data is a little bit different than the other data, and we were able to get a much larger sample size than the other projects. And this is because we only need photos, really. We don't need measurements. We don't need feather samples or anything of the likes. So we were able to use all of the photos of our live trapped individuals, as well as photos from eBird, which I'm sure everybody's familiar with. And then Also, we've gained photos from private individuals and something called VertNet. And so VertNet is really just an online repository of museum study skins and measurements and coordinates that we were able to use. And they even, most of the time, list the subspecies of the red-tailed hawk that they have. So we would use the ones from Illinois from that, and we got a pretty large data set. As far as identifying subspecies and determining the past distribution, we used raptor identification field guides from Wheeler and Liguori. And then once we had all of our data collected, identified, and the locations down, we were able to plug it into something called ArcGIS, which is a map making software, and perform spatial analysis on our data. So we'll get into a little bit about the different subspecies here. So this is the eastern subspecies, Buteo jamaicensis borealis. And this is a subspecies that is year-round, found in Illinois and in a lot of regions in the eastern portions of the United States, as you can see here. A lot of borealis, the eastern subspecies, also it spends their breeding time in eastern Canada and the northeastern portions of the United States, but they are year-round residents as well. The western subspecies, Buteo jamaicensis coloris, is a little bit different and has a little bit different breeding location. So they only are found wintering in Illinois. So they spend most of their year-round time in Western United States. You can mostly be found there, and they breed in Western Canada primarily. So they really only spend their wintering months in Illinois. So here's a combined map of the two more prevalent subspecies here, the borealis and the coloris. Although the coloris only winters in Illinois, Wheeler et al. In 2018, they had found that their distribution was statewide, so we went ahead and combined them on the same map. So if you can see here, the black dots are the coloris subspecies, the western subspecies, and the kind of grayer, lighter dots are the borealis subspecies. As you can see, we got a lot of samples in the Cook County area, and that's primarily from eBird. This still shows that these birds, both subspecies, are distributed throughout the state in the winter months. And so that's the key there, is that this is showing the winter distribution for the 2023. On the left, you can also see kind of the percentages of birds sampled. Obviously, a huge, significantly greater number of borealis sampled compared to the others. Our next subspecies is the Harlan subspecies, Buteo jamaicensis harlani. And this is, a diverse subspecies, but typically a darker morph. There are light morph Harlan's hawks as well, but these guys actually are known to breed all the way up in northern Alaska and northwestern Canada. So this is a really, really far migrator that can still be found in wintering months of Illinois. So there's distribution, however, is a little more limited. At least it was in 2018, where Wheeler described their location being primarily around the Mississippi River and kind of the habitats surrounding that, as well down into the Shawnee region there. So we had a smaller sample size of the Harlan's hawk, but we still were able to decide that their distribution is expanded east and north. So about 90 kilometers east and 70 kilometers north, and that's described by the sampling locations that we found. So one of the next subspecies is a much lighter morph, as you can tell, and this is the crider subspecies, so Buteo jamaicensis crideri. And these guys are known to breed in the central Great Plains region of North America, and as well in somewhat of central Canada. So they are also known to winter in Illinois, and their distribution is a little bit more expanded in 2018 than the Harlan subspecies. But again, we're proposing to expand the subspecies distribution about 97 kilometers east and about 44 kilometers north. Again, this is a relatively small sample size, but they're still suggesting that these subspecies are moving slightly. So the final subspecies that we wanted to talk about was the suggested northern subspecies. So, right, this is the proposed new subspecies that's kind of hotly debated by some ornithologists. And this is Buteo jamaicensis abieticola. And so as you can see on the right, this is where we found the northern subspecies distributed throughout the state, and it's likely a statewide distribution, especially in the winter time. As you can see, some sightings in the very southern extent and all the way in the west as well. So all of this subspecies data is super important especially to create kind of a baseline data set for us in the face of climate change and those sorts of habitat changes and any population changes that these subspecies may go through and shift their distribution even further. So that's sort of all we had today. And I want to give an extra special thanks to the DuPage Birding Club for making this research possible and having us out tonight to talk. And I think we'll go into any questions from the host or from the audience.
Yeah, if anybody has any questions, they can either add them to the chat or they can speak up. But yeah, thank you very much, Stephen, for that. That was very, very interesting.
Thank you.
Anybody have any questions about anything?
Yeah, Jerry Zaramorowski chatted, just comment that you didn't use gloves during your examination of the birds and was surprised by that. Can you comment about that?
Yeah, what style gloves were you kind of typically used to seeing, like plastic examination gloves or sort of protective gloves?
More protective.
Yeah, so that's actually a very interesting point, right? So we actually use a special holding technique that kind of isolates the talons and they're not able to use them or kind of come at us in any way. So by holding them sort of on the upper thigh, what we do is we'll have one finger in between to sort of support the legs and keep them from, pinching too close together. And then we'll have the other fingers wrapping around and supporting the bird upright. And so that really kind of just makes it look like the bird is perching there in your hand and has no room to get you at with the talons. And really the most susceptible time to getting poked is when we're transferring the bird between people or trying to take the measurements of the hallux or of kind of the upper leg. So, yeah.
Do you have a distribution or our percentage distribution of the various subspecies? What's your best guess on percentage of harlequin versus the ones that you mentioned here?
Yeah, so the eastern subspecies in Illinois is going to be by far the most prevalent. We saw probably 85 to 90% borealis subspecies here. And I think it goes down as you kind of get into those more rare subspecies. We saw one Harlan's hawk this year near Moraine View State Park. And so they're rare, but there's still that chance. I would say maybe, I think, what did we have on the one slide? So we had about 1.5% for Kreider's hawks, 2.5% for Harlan's, and then 5% for the western subspecies and for the northern subspecies. So, yeah.
Okay. Joe Suzeki asks, where does the suggested northern subspecies nest, and do they only winter in Illinois?
Great question. Yeah. So the northern subspecies is actually going to do most of their breeding in northeastern Canada and kind of the northeastern United States. So they're really in northern subspecies, and they are prevalent in Illinois, but they definitely can be found sort of throughout that range leading up to that area. And again, that's a sort of a proposed subspecies, and the identification of those can be difficult based on kind of their plumage being similar to the eastern subspecies, the borealis. Yeah.
And Donna asks if you could elaborate a little bit more about the isotope map and why do the isotopes vary as a function of latitude?
Yeah, great question. I think I skipped over that a little bit when I was talking about it. And so the Isoscape map is essentially when rain and precipitation is brought up into the atmosphere from the equator, it's going to have whatever the highest ratio of that heavy hydrogen isotope possible, right? And so as it's moving throughout the landscape, the heavier hydrogen isotopes are being deposited first and are kind of coming out in that precipitation first. And so as it moves across the landscape, you can actually see and test and measure that those hydrogen isotopes are becoming less prevalent. Those heavier isotopes are becoming less prevalent as you move north and as you go through that latitude. And it wasn't exactly, north-south. It kind of went following probably those weather patterns. But yeah, that's essentially how those work. So I hope that it's fairly complicated, and trying to wrap your head around it can be a little confusing, but I hope I did my best explanation of that. Yeah.
Yeah, Nancy asks, is it true that red tails are one of the few hawks that will attempt to attack a much larger prey, such as a blue heron?
That is an interesting question. I'm not convinced that they would have the gusto or the will to go after a blue heron, unless I'm not entirely sure about that one. I think they do tend to stick to small mammals, and they will go after birds, of course. They've been seen catching pigeons and slower birds. They're not the most quick, and I don't think that they're the most likely to go after that big of a prey. I'm not entirely sure, but they are opportunistic hunters, and so if there's an injured one or something like that, I'm sure they would have their way if possible. Yeah.
Which month do they nest in general?
Yeah, so I think that I must admit I'm not entirely familiar with that, and I haven't done. I should know that, but I want to say in the early April months, in the spring and summer.
Okay.
Just maybe refer to Dr. Harper for that.
Yeah, Stephen, do you also want to talk about watching the courtship display?
Oh yeah, no, yeah. So we actually saw we got to see a male and a female red-tailed hawk. This was fantastic. Yeah, so they do something called a sky dance, where they fly high in the sky and then soar back down and kind of are showing off to the female. And then essentially we saw them actually perform courtship, where they do their cloacal kiss and do the things they do. And after that, this male bird, after they did their thing, he came down and landed right on our trap, and we caught it right after the male and the female had mated. So that was kind of a fantastic kind of seeing the ecosystem at work there. It was neat.
Do you find that the year-round subspecies get into conflict with those subspecies that migrate here in the winter?
That's a good question. So red-tailed hawks are fairly territorial, and so they will mark out their kind of territory, from what I understand, and not necessarily defend it, but it's kind of just understood that they each have their kind of, the breeding pair has their own little region. And so I would imagine that if these red-tailed hawks, different subspecies, are returning back to the same area each year, that they probably aren't coming into too much conflict. And however, this may be different as climate change is pushing birds, to different locations, and it causing these kind of interspecies interaction, or interspecies interactions. Yeah. Competition.
Speaking of climate change, do you find that that's affecting their nesting success with the shifting of the spring getting earlier?
Yeah, that's an interesting question too. And I think it's not only the spring getting earlier, but the more severity of weather, frequency of severe weather and cold snaps being more irregular and being not as easily detectable and different things like that. So I think that probably will inevitably be an impact to red-tailed hawks, and that may be an area of research that to continue is having a baseline, what factors play a role in the success of red-tailed hawk breeding. Dr. Harper, did you have something to add to that?
So no, my perception is that we're seeing more sort of higher wind events, 20, 30, 40 miles an hour. And I'm not sure how this will impact breeding, but certainly, with these higher wind events, you would think perhaps you may have more tree falls, but I'm uncertain.
I think we have exhausted our messages from our members right now. Robert, you want to bring it on home here?
If I could actually say one more thing. So I appreciate very much your generous donations. I appreciate what you're doing, trying to get people interested in birds and out in the natural world. I think you have a very impressive organization. My students, actually Leah Beineck, she gave a presentation to you in person pre-COVID. That was in the fall of 2019. And I just want to say how much we appreciate your generous support.
Yeah, well, thank you very much for educating people on this, and we're happy to have you and your students back anytime. So if that's it, if nobody else has any questions, again, thank you, Stephen. Thank you, Dr. Harper, for joining us this evening. Thank you, everybody else, for logging on as well. And we will look forward to seeing you at the next meeting, which is on May 11th. So thank you all very much, and I hope you have a good evening.